Well, Michelle, thank you very much for that very generous introduction. I've always had a connection with LA. Growing up in Brooklyn, my dad took me to see the Brooklyn Dodgers. And I think in '57, the Dodgers left to come to LA. Most people in Brooklyn hated the Dodgers after they left Brooklyn. But I was only 5 at the time, and for some reason I never got that passion, so I still am a Brooklyn Dodger or LA Dodger fan. Also, growing up in Brooklyn, I tend to speak fast, and lots of people think that everyone in Brooklyn speaks fast. That's really not true. There are people that are born in Brooklyn that speak slowly. They just don't survive. So if I'm going too fast, please raise your hand, and I'll be happy to stop. I also have a little cold, so I apologize for that. So the work I'll present today is more of a basic science project, but really it's integrated with the Wellstone Center at the University of Iowa, and I'm lucky to have Kathy Matthews and Steve Moore as colleagues that help run this center. We actually started working together around 1995, just meeting to discuss muscle biopsies that were coming into my lab or into Steve's pathology lab. And around 2005, we put together a grant, and we've been funded since then, and it's really been quite nice to have this interaction. One of the main components of the center is we have a monthly meeting where we discuss new biopsies, and every year I think it's going to end, and we're going to have, there's not going to be any new patients coming along, and that's really not the case. It just continues and continues. And what's surprising, we're getting a lot of cases now involving the ryanodine receptor, sodium channels, all these proteins that I worked on years ago that I thought would never be involved in muscular dystrophy or muscle disease, and so that's quite interesting too. I'd like to acknowledge the people. I'll be presenting work from all of these people during my talk. The one thing to note on this slide, these are former postdocs, is that they all have jobs, which is successful, except Amaya, who's a new grad student in the laboratory. The undergrads in our lab really contribute an awful lot, and we recruit undergrads from chemistry, engineering, various fields, and it ends up, surprisingly, a lot of them now are going into science. So even though they're around a lab where people are getting kind of depressed with the funding situation, they get excited about the science. This is a good example. Ellie, when she was a senior undergrad in the lab, if you had a genotyping problem, you went to Ellie, no matter who you were. She was really good. She just got her PhD with Bob Dirksen, and now she's going to be working with Charles Thornton at Rochester. So they've really done really well. Zihan, when Yuji left the lab, she kept the project going for a year between Iowa and Japan just by doing the work. So really, it's really fun, like probably it is here, having undergrads involved in the research, and they work with us up to 3 or 4 years. So the lab is funded from HHMI really to kind of do more basic science on trying to understand the cellular and molecular mechanisms underlying muscular dystrophy. And we also are funded from the Wellstone Center for more translational research projects that we have with Steve and Kathy, aimed at diagnostic work and also aimed at trying to develop therapies. So for today, I'll first just give you a brief introduction to Dystroglycan, then discuss some of the molecular pathogenesis of the dystroglycanopathies, and then discuss the novel post-translational processing and glycosylation required for dystroglycan's function, and then end with the structural basics of dystroglycan function as an ECM receptor. So muscular dystrophy is a term that covers a large group of diseases that are genetically determined, where the primary defect is in skeletal muscle. And for a number of years, tissue like this was studied, where you have muscle cell death, atrophy and hypertrophy, fat infiltration, fibrosis, and it really was quite difficult to identify what was the cause. And if you look back at the older papers, there's many different reported causes of muscular dystrophy. And then Lou Kunkel, Kay Davies, and Ron Wharton studied the gene, and Lou Kunkel discovered the dystrophin gene, and him and Eric Hoffman discovered the protein dystrophin. So that was the beginning of the genetics of muscular dystrophy, at that time called using reverse genetics. Since then, and this slide is outdated, the number of genes involved in causing muscular dystrophy has dramatically increased. And one interesting fact is if you look at those gene products, around 70%, but not all of them, but around 70% are found at this interface between the muscle sarcolemma and the muscle extracellular matrix. So there are proteins like dystrophin, which is in the cytoskeleton, proteins like sarcoglycans, which are in the membrane. There's proteins like collagen or laminin that are in the extracellular matrix. And I'll be telling you about enzymes that actually process dystroglycan that's sent to this interface. So this seems to be a hot spot for the muscular dystrophies. But again, there are other proteins that are, contractile proteins and calpain-3, which are not found at this interface. As Rochelle mentioned, our work started with looking at this complex of proteins associated with dystrophin. To be real honest, we got involved in the project because we thought dystrophin might be involved with the calcium channels we were studying. It really wasn't a planned project. It was a whole summer of studies where we kept getting disappointed. The calcium channels and sodium channels were purifying away from dystrophin, and we ended up with this complex of proteins. It's still just a cartoon model. We've tried to get some EM structures and still have not been successful, but basically this is a cartoon model of what we think the complex is doing. Dystrophin interacts with F-actin underneath the plasma membrane. It interacts with a group of cytoskeletal proteins, transmembrane proteins, and the main protein we've been studying is dystroglycan, which links the dystrophin C-terminal through the membrane to the extracellular alpha-dystroglycan. This is a non-covalent strong interaction. It's heavily glycosylated, and it binds laminin G-containing proteins, such as laminin, agrin, and perlecan in the extracellular matrix. So one function when we first described this was that this complex plays an important role linking the basement membrane or extracellular matrix across the plasma membrane to the actin cytoskeleton. Now, in Duchenne muscular dystrophy, and this is actually how we got started, when we had this complex picture from biochemistry, we really wanted to look at a case where dystrophin was missing, and the mouse wasn't available at the time, and so we went to the neurology, peds neurology, to get samples which were missing dystrophin. And if you look at Duchenne muscular dystrophy, which is defined by the absence of dystrophin, the other components of the complex showed here, sarcoglycan and dystroglycan, are dramatically reduced. So it looks like they're present, they're made, but they probably get degraded or not properly trafficked to the plasma membrane. And so this suggested that almost the entire complex was lost in Duchenne muscular dystrophy. Now, since then, our lab and lots of labs around the world have found that this complex itself is a hotspot for muscular dystrophies. So Duchenne-Becker caused by the abnormalities in dystrophin. The sarcoglycan-sarcospan complex can lead to limb-girdle muscular dystrophies. If you're missing laminin 2 or the merosin, the muscle form of laminin, can lead to a congenital muscular dystrophy. And then there's a growing group of diseases where the protein dystroglycan is present but is not properly processed. And those are the diseases called dystroglycanopathies that I'll be discussing today. So we became involved in this study when we were sent samples of muscle-eye brain skeletal muscle or Fukuyama congenital muscular dystrophy. And when we stained those biopsies with antibodies to beta-dystroglycan, you saw a nice staining. This is from a very young patient, so the muscle cells are quite small. When we stained them with two antibodies that we knew recognized the glycosylated form of dystroglycan, there was almost no staining. But if we had a core antibody staining, core antibody that could actually stain these biopsies quite nicely. So this suggested that the glycosylation was abnormal, but the protein was present. In the cases where we had a large enough biopsy, we could do Western blots, and you can see beta-dystroglycan is normal, but the antibody 2H6 is deficient, but the core antibody shows the protein is present, and it's around 60 kilodaltons, smaller in molecular weight. Now, we knew from the early studies that glycosylation was important for binding to laminin, and this is a case for Fukuyama congenital muscular dystrophy. If you look at high-affinity laminin binding, it's around 80 or 90% reduced, suggesting that the absence of this glycosylation led to a non-functional dystroglycan. Around the same time, Dan Michael in the lab had found this mouse, which had been at Jackson for a long time. The staining for 2H6 is completely deficient in this mouse, has the same type of shift, reduced 2H6 staining, reduced laminin binding. So this mouse gave us an initial mouse model. Now, most of the congenital muscular dystrophies have a severe CNS abnormality, and so the mouse gave us a chance to look at the brain, and so you can see, again, in MYD skeletal muscles, around an 80% reduction in high-affinity laminin binding, so there's still integrins present, but a dramatic reduction. And in brain, it's almost like a 95% reduction in high-affinity laminin binding, suggesting that dystroglycan, we knew was present in the brain and bound laminin with high affinity. So in general now, normal skeletal muscle has this complex that links the cytoskeleton to the extracellular matrix. In Duchenne and limb-girdle muscular dystrophy, in the models, this complex is disrupted in various ways, and you don't link the cytoskeleton to the extracellular matrix. In this group of dystroglycanopathies, the entire complex is present. You can isolate the complex, it's still intact, but it lacks the link into the extracellular matrix and leads to muscle disease. So the working model for several of the muscular dystrophy genes, and again, not all of them, is that there are a group that are associated with the dystrophin glycoprotein complex that we believe really prevent structural damage to the sarcolemma membrane. And there's a group that we've studied and other groups are studying which really are involved in repair of the sarcolemma membrane if it does get damaged during muscle contraction, and that's associated with dysferlin. And we're currently actually not working on this, but it's a very interesting mechanism. So dystroglycan is a novel extracellular matrix receptor. It's widely expressed. It's expressed in all cells. It's even expressed in T cells. It binds laminin G domain containing proteins such as laminin, agrin, and perlecan, but this group is actually growing. There's more ligands coming out each year. It maintains and organizes the basement membrane by binding these ECM ligands with high affinity. It's involved in various developmental and physiological processes, especially in muscle and brain. But one unique aspect is it requires extensive glycosylation, and it's up to at least now 18 genes involved in this processing to really produce a functional extracellular matrix receptor. So I'm going to first tell you this about the processing of dystroglycan from the protein level. So it's produced as a pre-pro dystroglycan with a single peptide and a single transmembrane domain. It cleaves itself into alpha and beta dystroglycan. And then the beta dystroglycan stays in the membrane, is glycosylated, and is the link into the dystrophin or utrophin molecule. It covalently is associated with alpha dystroglycan, which first starts off with an N-terminal domain, a mucine domain, and a C-terminal domain. The N-terminal domain, I'll show you in a moment, is important for the glycosylation. The mucine domain gets totally covered with sugars, mostly O-linked sugars. If you do peptide analysis, you find almost very few peptides that are not glycosylated. And then during processing to the surface membrane or at the surface membrane, furin cleaves the N-terminal domain, so this is removed. It's not present in the mature molecule, and you're left with alpha dystroglycan here, which has extensive glycosylation and covalently associated with beta dystroglycan. So its processing by itself proteolytic processing is quite complex. The initial two glycans that were found from an analysis of alpha dystroglycan were a mucine glycan and a mannose glycan. We think these are quite important for the structure of dystroglycan, but we don't think they're directly involved in laminin binding, and I'll show you that in a moment. As I mentioned, the dystroglycan binds to these LG45 domains on laminin and also LG domains on these proteins. If you look at Western blots of various tissues with an antibody that recognizes the glycosylation, you can see the molecular weight is really quite different. In brain, it's almost like 110, 120 kilodaltons. In heart, it could be up to maybe 180, 190 kilodaltons. So it has tissue-specific glycosylation. And all these forms of dystroglycan bind laminin with very high affinity. Now, dystroglycan has been implicated in diseases. The one we'll discuss today is the dystroglycanopathies, and this is this group of diseases where you have disruptive glycosylation that really you lose the function as an extracellular matrix receptor. We have had a long-standing collaboration with Michael Oldstone down at Scripps, and he found that dystroglycan was the receptor for old world arenaviruses, such as Lassa fever virus. And it's interesting, the same glycosylation is required for the receptor function for the virus. So without glycosylation, the LCMV virus or the Lassa fever virus doesn't bind to dystroglycan. And there are also cases of fetal LCMV infections where you lead to abnormal brain development. So most of the time in adults, fetal or LCMV infections, this mouse virus infection really doesn't cause any problem, but if a woman is pregnant and she gets a severe infection, it could cause abnormal brain development. And we think it's by competition with dystroglycan during brain development. And finally, another story is epithelial cancers. So in many epithelial cancers, the genes involved in the processing of dystroglycan are silenced. You get this hypoglycosylation phenotype like you find in the patients, but the exact physiological and importance of this whole process is really still not known. We have a few studies on it, but it's quite interesting in terms of the hypoglycosylation, but we really don't know what role it plays in cancer. Now, the dystroglycanopathy genes, there were originally 6 found. As I showed you in that first slide, we looked at 2 of those. And most of the initial cases were in Walker-Warburg syndrome, which is a very severe congenital muscular dystrophy. The children die in the first few days or weeks of life. They have very severe muscular dystrophy and a severe CNS phenotype. And at first, people thought that's the only kind of phenotype you would see. But then as more and more cases were found, a milder phenotype, muscle-eye brain, was found, and CMD without brain involvement, and finally, limb-girdle muscular dystrophy. So we now know that really these genes can cause a spectrum of disease from very mild limb-girdle dystrophy to really severe congenital muscular dystrophy. And the initial 2 genes, the function was POMT1, POMT2 forms a complex that adds mannose onto this mannose glycan, and then POMGNT1 adds glucnac. And so this was the initial findings. So these 6 genes we were identifying and working at in our CM Wellstone Center. And around 2005, it seemed that new gene discovery leveled off. There weren't any new genes coming along, although from the muscle biopsies, around 50% of the cases we were getting were dystroglycanopathy. So they were deficient in glycosylation, but no genes were found. And this is where the center really came quite useful. We have this monthly meeting, and part of this meeting we discuss biopsies, and one of the cases we discuss are cases that have both muscle biopsies but also skin biopsies, and that they're deposited in this core that Steve Moore runs, and actually he sends out lots of samples to various investigators. So this core was quite valuable, and in the core we had lots of muscle biopsies, around 100 fibroblast cultures. And so Toby Willer was a new postdoc in the lab from the yeast field. And most people from the muscle field would have never tried this experiment, but he didn't really know better. He said, why don't I look at skin fibroblasts if they have dystroglycan? And he found that wild-type skin fibroblasts from normal humans have nice dystroglycan. It's glycosylated, binds laminin. And so this was really quite nice. And then he quickly tested skin fibroblasts from 6 patients with these severe Walker-Warburg phenotype. And there's no glycosylation, no laminin binding, but the protein is still produced. One protein shifts in molecular weight. We're still working on that. But the protein is produced just like you find in the muscle biopsies. And so this was very exciting for the patients, because now you could skip the muscle biopsy and do the skin fibroblasts, and that's what we do now. And also you could quickly identify which gene was causing the disease. So again, Toby coming from the yeast field developed complementation assays, and this is complementation with a patient with a POMT1 mutation. He infects the cells with adenoviruses expressing the different genes, and POMT1 rescues the glycosylation and laminin binding. And so he could then do this experiment first, and then we only had to sequence 1 gene instead of sequencing all 6. He also developed an on-cell assay with essentially the same results, just looking at the cell surface staining. Again, coming from the yeast field, in yeast you can do a yeast mating assay where you fuse 2 cells. He developed a polyethylene glycol fusion assay. So this is a patient with a POMT1 mutation, no staining, fucotin, no staining. But if you fuse these 2 cells together, the Golgi mixes and you get nice glycosylation of dystroglycan. So that gave him a tool to screen for new genes. So he took all the patient cells we had and did this massive fusion assay and came up with 5 new genetically distinct groups that were new genes. And then using exome sequencing and actually in collaboration with the group here, Stan's group, we're able to identify these 5 new genes. Now, one thing nice about having this assay, when you do exome sequencing, sometimes it's difficult to really nail down that this is the gene that causes the mutation, causes the disease. But here we could do that nicely. So with this gene ISPD, we could show that the wild-type ISPD would rescue. You can see rescue laminin binding, rescue 2H6 staining. So it was very nice. The mutant gene did not. So this enabled us to really discover a large number of ISPD patients. It's interesting, each center kind of collects their own set of patients. We were getting more patients in the phenotype of the congenital muscular dystrophies, and so we thought ISPD was only a gene involved in congenital muscular dystrophies. We shared the information with Francesco Montoni, and he pulled up all these limb-girdle muscular dystrophies. So it's actually a gene involved with a lot of limb-girdle muscular dystrophies too. So since that took off, and especially with exome sequencing, again, it's not just our lab, it's many labs, and we're up to around 18 genes involved in the processing of dystroglycan. And I think it's probably going to go up at least another 6 genes over the next few years. So it's really a question of why you have all this energy put into this glycosylation story, and we still don't have an answer for that, but it's really quite complex, and it shows you, why the genotype-phenotype correlation is so important. So this is an example of one of these new genes, TME5. It's a case with a severe muscular dystrophy, no 2H6 staining. So all those 18 genes give the same phenotype on muscle, the same biochemical footprint, no 2H6 staining, no laminin binding in the severe case. So we were left at this point with these relatively unknown genes. A few of the genes, we knew their activity. We had some biochemistry data. This was the original glycan found on dystroglycan. There was some biochemistry data on dystroglycan that there was a phosphorylated mannose glycan. And really, we really didn't have any hint at the function of these new genes. So to begin this study, we actually first did a study on a patient biopsy that came from Turkey. A colleague in Turkey had identified a patient that had a dystroglycanopathy, and he had sequenced and found that there were no mutations in the known genes. And he sent us her muscle biopsy, and we did DNA, and we sequenced, and we actually found the mutation in dystroglycan. And this was quite surprising, since the mouse model of knockout of dystroglycan is lethal, and a few patients that have been born die in the first few days of life. So we thought we would never find a mutation like this. More surprising, it was in the N-terminal, which I said is not present in the mature protein. So this is a quite interesting case. We made dystroglycan null myoblasts and then expressed the wild type and the mutant dystroglycan in those myoblasts, and found that they mimicked what you saw in the biopsy, that there was a decrease in glycosylation with this mutation and decrease in laminin binding, suggesting that really this wasn't a glycosylation site, but it was involved in regulation of glycosylation. Now, we had some earlier data suggesting that the N-terminal domain was involved in the enzyme Lodge, and this just shows that if you pull out Lodge with anti-MYC associated with the N-terminal domain here, 2 different constructs, you get nice binding of Lodge to this DGN, the N-terminal domain. But the mutant DGN, where that mutation is, has dramatically reduced binding. So we went on. We had this project, kind of a draft manuscript, and I told the postdoc to make a mouse, and he asked me, do I need the mouse for the paper? And I said, no, you don't need the mouse for the paper. But I knew we needed the mouse for the paper, but I just couldn't tell him that at the time. But he went on to make the mouse. It actually worked really well, and the mouse has the same phenotype as we saw in the patient, this reduced staining, reduced laminin binding. It's interesting that the phenotype of the patient has a more severe CNS phenotype, and the mouse also has a more severe glycosylation defect in the CNS. And the mouse develops a mild, like limb-girdle muscular dystrophy like the patient has. So this is our working model, and this gave us our first hint towards the glycosylation story, is that in wild-type Golgi, the DGN, the N-terminal domain of dystroglycan, is very important to target Lodge to the dystroglycan molecule for that final step in glycosylation, and then it's trafficked, and the mature molecule doesn't have DGN, but is fully glycosylated. In this patient, this interaction is greatly reduced, so you have incomplete glycosylation, and so you have a dystroglycanopathy phenotype. Yuji has since, and I don't think we've published this, found a patient with a Lodge mutation where the same thing is true. This disrupts this interaction between the DGN and the Lodge enzyme, and you end up with a hyperglycosylation. So this suggests that really the N-terminal domain is very critical for this final step in glycosylation. So the next enzyme we went to work on was Lodge, and having this hint that it was involved with the N-terminal domain. We also knew that if you overexpress Lodge, you got increase in staining with this enzyme and increase in laminin binding, suggesting that it was really critical for the laminin binding. If you do this experiment with several of the other enzymes, you actually decrease some of the staining. So Lodge was a gene that was originally named Lodge because of the fact that it was so big. It's really not that big compared to dystrophin. And there's a spontaneous mutation in Lodge that produces that mouse that I told you about, the MYD mouse. It's kind of an interesting gene in that it has two essential domains that appear to be glycosyl transferases. So it looked like a bifunctional glycosyl transferase. And we decided to see if we could make Lodge and have an active molecule. And this is the best enzyme out of these 18 that we've worked with. So we removed the transmembrane domain and tagged it. And we could produce the protein quite nicely. And this is Kamasi blue gel. So we're now trying to do cryo-EM on Lodge, but it really works very well. This similar experiment with half of those genes doesn't work at all. Golgi enzymes are kind of difficult most of the time to work with. But this enzyme does a really nice job. We then did an experiment. We had the enzyme, but we really had no idea what sugars were involved. So we overexpressed Lodge in cells and then isolated dystroglycan, alpha-dystroglycan, and then did sugar analysis. So two sugars pop out when you overexpress Lodge, xylose and glucuronic acid. So that gave us our first hint that these were possible substrates that Lodge was using. And then we developed an assay, a simple assay, where we took a fluorescent xylose molecule, the Lodge enzyme itself, and added all the sugars that we could get our hands on. You have to use the activated sugars, UDP sugars. And with UDP glucuronic acid, we got a product, and with mass spec identified that as glucuronic acid being added to xylose. So this gave us one of the enzymatic activities for Lodge. And then we took glucuronic acid that was fluorescently tagged, delta transmembrane Lodge. We got another product with UDP xylose, and that gave us this step. So really, Lodge was this bifunctional glycosyl transferase. By doing mutations in these DXD motifs, we found that this first domain is a domain for xylose transferase activity, and the second domain is for glucuronic transferase activity. So this defined the two enzymatic activities. Now, in those early gels I showed you, you see the dystroglycan, alpha-dystroglycan is a very broad band, and it's because it has this polysaccharide on it, which we now call matriglycan. But if you take both sugars and do this assay, you get many peaks on the HPLC, and you're actually producing this polysaccharide of xylose glucuronic acid repeats. So this enzyme can go on and on. We don't know the results or the total number of repeats in the native molecule. We think it could be between 50 and 100. So it's really an extended polysaccharide that's added on to dystroglycan. We could also take these polysaccharides and isolate them on HPLC, and the smaller ones we could analyze with NMR to show the exact structure of the molecule. So now this is a summary of this glycosylation. I've only really told you about one enzyme so far, but this is what's known from both our lab and other labs. So initially, POMT1, POMT2 in the ER adds mannose. There's probably 2 sites on dystroglycan, but that hasn't been fully clarified. And then POMGNT2 acts, they add glucnac, and then B3-galNT2 adds galnac. And then this trisaccharide is phosphorylated. I'll tell you a little bit about that by a novel enzyme called POMK. And then recently, Toda's group in Japan showed that fucotin and FKRP are able to add this ribitol 5-phosphate to this position, to galnac. And then our group and the Japanese group showed that TME5, that enzyme, that patient I showed you earlier, has xylose transferase activity, and it adds it to this ribitol. This structure stuff is not fully clarified, and the enzymatic activities of these two proteins are still it's really not that strong, but it looks from the mass spec data that they're probably correct. And then B4-GTA1 adds glucnac to xylose. And we think this disaccharide is really the primer for Lodge. It has a beta-1,4 linkage, and then Lodge acts on this with a beta-1,3 linkage to produce the polysaccharide of glucuronic acid and xylose. And so that's defining most of the enzymes. Some of the other enzymes are involved in producing substrates for this. ISPD is involved in producing a substrate for this reaction. There are substrates produced for the mannose. So the other enzymes are feeding in at an earlier step in substrates for these enzymes. The POMT1, POMK enzyme is quite interesting. It was originally called SGK196. And Tony Hunter had a map of kinases that he produced in this paper in Science in 2002. And SGK196 here was thought of as a pseudo or a dead kinase. It didn't have the proper structure actually to look like the A kinase. We had nice rescue data showing it had activity. We had this nice assay where if you take mannose that's dislabeled with a fluorescent tag, plus or minus ATP, there's no product. The disaccharide, no product. But if you take the trisaccharide, you get a nice phosphorylated product. So our data was pretty strong that this was the enzyme involved in the phosphorylation. The reviewers of the papers made us kind of add a comment that this could have been like a coenzyme or a pseudoenzyme attached to the real enzyme. So then we went on with Jack Dixon to explore this. We did some nice binding assays with the trisaccharide with our pure enzyme and got high affinity bindings with this trisaccharide, suggesting that it was really binding the substrate. And then with Jack's lab, we were able to crystallize. This is I think the just zebrafish palm K. And it looks just like the A kinase. It has the same structure, even though it doesn't have the so-called 6 or 7 amino acids that are required to form that structure. But both our lab and Jack's lab never thought about the fact that this kinase is actually in the ER. So it has 3 disulfides shown here. So it pops into the same structure. It looks like the A kinase, but it does it with a totally different mechanism with disulfides. And Jack is really excited because there's around 100 pseudokinases that people think are dead kinases because they don't configure like the A kinase. But if they are within the lumen of the ER or Golgi, they actually could obtain the same structure using a totally different mechanism. So this is exciting for the kinase field, but it's also interesting that we think it's just that single protein that really causes the phosphorylation of the mannose. And as I just mentioned, this group of enzymes is located in the ER, and this group of enzymes are in the Golgi. The current genes that we're working on, although there's not a lot of patient data on this, are genes that traffic proteins from the ER to Golgi. And we think those could also lead to dystroglycanopathies, it's possible that in some cases the trafficking is not correct or the protein is trafficked too fast or too slow. We also think the glycosylation, the extent of glycosylation might depend on the time it actually spends within the Golgi. And so trafficking can also probably cause problems in the extent of glycosylation. So that's the current status of where we stand in terms of the glycans. Now, everything I told you about in terms of the glycans are glycans that we are synthesizing in the lab. We really didn't have strong data that the native molecule had this glucuronic acid xylose repeat. And Takako Moroguchi was able to isolate 2 enzymes, one that could remove xylose and one that could remove glucuronic acid. And if you digest the native dystroglycan with each of these enzymes by itself, they don't really change the molecular weight at all. But if you put both enzymes, you totally remove this laminin binding and the 2H6 staining. So this experiment says on the native dystroglycan there is this repeat, and that repeat is what's really responsible for the high affinity binding and the binding for 2H6. Now, using these 2 enzymes, if we used one at a time, we should be able to see how many repeats are on dystroglycan. But so far, I haven't been able to get any postdoc in the lab to try that experiment, doing one digestion at a time. Maybe some new postdoc. Usually, they listen to you for the first like 2 or 3 weeks when they're in the lab. And so they won't know better, and they'll try it. But that's something we're really very interested in determining. I'll tell you why in a moment. So now we can start to produce these glycans. And when we produce them, we can either get the low molecular weight up to maybe what we call G10, 10 sugars, or high molecular weight better than, say, G15 or 20 sugars in a row. And when we did binding on the pure glycan, we could see that the pure glycan can bind laminin with very high affinity. So this demonstrates there's nothing with the protein. It's really the glycan that binds laminin. So that was quite exciting, and most people didn't think that the glycan could have this high affinity. But the low mass sugars really were just barely above background, so we weren't sure about the low mass sugars. And that was kind of confusing. Same thing with this antibody 2H6, the pure antibody and the pure glycans with high mass sugars bind very nicely. Low mass sugars just a little bit. And so we now know the epitope for this antibody that's used by us and lots of labs around the world for these patient studies. It's really this repeat of xylose and glucuronic acid. Now, if you look at the structure of the LG domain, and this was done by Erhard Hohenhester, there's a face where they believe that dystroglycan would interact. But if you look at this face, it's not that big. And why you would need 15 sugars in a row to bind there really didn't seem to make sense. So we decided to take another stab at this and see if we could obtain the structure of the LG domain with the actual sugars. So to do that, we had to start becoming a real glycobiology lab to make these defined sugars. And so we start off with glucuronic acid and add xylose and then purify the product, actually add new enzyme and then do the next step. The enzyme binds the sugar, so you can't keep the same enzyme at each step, and you end up with a defined product. So G3 is with 3 sugars, G9 would be with 9 sugars. We could use those enzymes that Takako had isolated to confirm the sugar structure, so we can take something like G5 and a single step at a time move it down to the single sugar. Excuse me. We could then do binding, and this is with G5, the first studies we did, and with an NMR approach where we're adding in the laminin G domain, the purified G domain, and we come up with a very high affinity binding, 0.23 micromolar binding. And laminin requires calcium to bind the dystroglycan, and in the absence of calcium, there's no binding at all. So this confirmed really that it was quite small in terms of the number of sugars required. We went even down further, and even with just 2 sugars, we could get fairly good binding, suggesting that it really was 2 sugars that made up the binding site. We then teamed up with Earhart and David Briggs, and we made enough of the glycans that they could actually soak it into the crystal. Before, we thought we might have to do co-crystallization, but what we did was we did crystallization without the glycan, got an image, and then put the glycan in. It soaked in quite nicely, and the different image is shown there. And when you model it, you find it's xylose glucuronic acid that are chelating the calcium that's on laminin, and this also interacting with the protein. And it's this disaccharide that forms the high affinity binding site, which was quite exciting to find. Now, there are other proteins that have laminin G domain containing G domains that bind to dystroglycan. And when you look at all those proteins, they have these conserved interaction sites, the amino acids where they interact with the disaccharides, suggesting that they bind with the same mechanism to this glycan. So in summary on LARGE, LARGE is a bifunctional glycosyltransferase that has both xylose transferase activity and glucuronic acid transferase activity. It produces this protein, this glycan called matriglycan, which is a polysaccharide of repeating units. And it's really this single disaccharide derives that binds directly to the ECM ligands. Now, if you look at the LG4-5 domain, there's an interesting question here. This is the domain that binds to the dystroglycan or to the glycan. But however, there's another site on here which so far has not been reported to bind dystroglycan, but it's possible that this is the high affinity site, and if the glycan's long enough, it could actually reach over to here, and essentially the affinity would even be stronger because you'd have 2 binding sites on the LG4-5 domain. So we're currently making a molecule called G20 that we think should span this to see if that's actually the case. But that would be quite interesting because that would really dramatically increase the affinity if it actually had 2 binding sites. Now, when we started this work, we were aimed at getting this structure. We always had questions at seminars, is it really the glycan? Lots of people wanted to think that the glycan was modifying the protein structure, and it really was the protein interacting with laminin. And no one really wanted to believe that it was the real glycan that would interact. And so we kind of really wanted to get and nail this question, but we never thought it would actually open up new questions. And when you think about it, if this is one site and the glycan is 100 repeats, then it's possible it's interacting with other proteins. And we actually went back to our old data, and we have evidence for that. So if you look at embryonic stem cells, they express dystroglycan very nicely. This is mouse embryonic stem cells. If you add laminin 1 to the culture, you get clustering of laminin on the cell surface. This is like the beginning of a formation of a basement membrane. It's quite different than the fibronectin matrix. And if you look at dystroglycan null cells, there's no dystroglycan, and in this assay, no laminin 1 binding. And so this is, again, thinking about maybe dystroglycan is actually clustering laminin because it can interact with multiple laminin molecules. That the binding site's a disaccharide, but the length is so long that it can interact with multiple laminins. Another place to look at this is in the brain. This is a mouse where dystroglycan was knocked out specifically in the brain with a GFAP-Cre with a flox dystroglycan. And this is the glial limitans, which has lots of laminin in it. The radial glial come up to the glial limitans. And in the absence of dystroglycan, you disrupt this basement membrane structure, again suggesting that maybe it's the multiple interactions that help stabilizing that basement membrane structure. So our working model is that the large glycan It could be long enough to maybe interact with multiple laminins or agrin and perlecan and really stabilizes the basement membrane structure, at the same time forming a link across the membrane to link the cytoskeleton into the extracellular matrix. And in the dystroglycanopathy cases, this glycan is either absent or greatly reduced, and you have a loss of this link across the membrane and maybe have disruption of the basement membrane. And that's our current kind of working model. Some further evidence from that comes from earlier work where we looked at the basement membrane in skeletal muscle. So in wild-type muscle, you have a nice tight association of the basement membrane with the sarcolemma. If you look at integrin 7 knockout mice, which have a relatively mild phenotype, probably more myotendinous junction phenotype, but they have a real nice tight association with the sarcolemma and the basement membrane. But in dystroglycan knockouts, you find many regions where you have the separation of the basement membrane from the plasma membrane without that tight link of dystroglycan. In some cases, the sarcolemma has been lost, but you still have the basement membrane. It's interesting, Andy Engel does a lot of really neat muscle pathology studies and also EM studies. And years ago, he found patients that looked like this. And then later, he found that those were dystroglycanopathies. So it really holds for patients, too, that you really need dystroglycan to form this tight association with the basement membrane. So this suggested an assay that Renzi Han developed in the lab, where we would look at muscle after being damaged. So in normal skeletal muscle, you have many links, and we think in adult muscle it's mostly dystroglycan between the basement membrane and the plasma membrane. And then when muscle's damaged, it could repair itself, possibly through that dysferlin pathway. But in the dystroglycanopathy cases, or in the absence of dystroglycan, the number of links to the basement membrane is greatly reduced. And when membranes are damaged, this damage can expand and maybe lead to muscle membrane rupture and muscle necrosis. So to look at this, Renzi developed this laser dye assay. And on top, I'll show you first in wild-type muscle, where he damages it with a 2-photon laser and lets this dye FM143 enter the cell. And you can see the dye enters, and there's a small amount of dye, and probably repair is taking place after that. Now, this is in the MYD mouse, which is missing that large glycan, the same laser pulse. You can see dramatically more injury occurs in the absence of that link to the basement membrane. So we think dystroglycan is really acting as an adhesive that allows the basement membrane, which is really much stronger than the lipid bilayer, to prevent the rupture of the sarcolemma. And we're currently trying to test this, and we're currently making glycans that we can add back to these physiological experiments to see if we can reform this link and what it would do. And this is really a principle of fraction mechanics, and you've probably seen this in high school physics, where the balloon you blow up is like the plasma membrane, and I'm going to put a piece of tape on the balloon, which is like the basement membrane. And if you do that, you can actually put a pin through the balloon. So there's the tape. That's my mutant finger. And there's the pin. And the balloon doesn't blow up because the tape is providing the strength there. You pull it out, it still doesn't blow up. You put it inside, it blows up. So we think that really the dystroglycan is acting as this adhesive, and that is really quite important for maintaining the tight association of the basement membrane and skeletal muscle with the plasma membrane. So in general, the dystroglycanopathies are this group of diseases where you have abnormal glycosylation leads to this loss of the link between the extracellular matrix and the plasma membrane. And this is the pathogenic mechanism, the underlying loss of glycosylation. And there are several different therapeutic approaches. The typical one would be gene therapy, which groups are trying, and it has all the potential and problems that you have with gene therapy. It might be a little bit easier because the Golgi enzymes are present at very low levels, so you're not going to need a lot of protein like you might need for dystrophin. However, what's more exciting, I think, for us and some drug companies, that when you have enzymes, and this is the first group of enzymes involved in muscular dystrophy, it gives you the possibility of doing therapies based on pharmacological approaches and such as glycotherapies or ways of stimulating these enzymes. all the enzymes have some activity in patients. So if you have a null in any of these enzymes in the mice or patients, it looks like it's lethal. So in order for a patient to be born, they have to probably have 5 to 15% activity. And hopefully that there might be ways to develop therapies where you stimulate that activity using small molecules to increase the glycosylation. In particular, the enzyme largeware might be an exciting one to work on, and that's something we're currently considering. So just to thank the collaborators, I think I mentioned most of them during my talk. To thank the current group of people in the lab. We now have a coffee house in our building, so they're quite excited. And Howard Hughes gave us this nice black T-shirt. And Takahiro is a new postdoc in the lab and was very excited about the T-shirt and wanted one for his wife. And so he asked my administrator if he could have an extra T-shirt. And I told my administrator, well, go back and say that, you know, once his paper was accepted in Science, that he could give the T-shirt to his wife. And this is our recent Halloween party. I'm showing you this because these are my 2 grandsons. Finally, you know, at our conference every summer, we have a T-shirt that we have. And in the past, sometimes we've had white T-shirts, and white kind of resembles the color of my hair. But last summer, we decided to go with a new color. And if you happen to recognize this color, it might resemble, you know, somebody's hair. So this is a gift for Rochelle to go along with her red hair. Now, Rochelle doesn't know this, but you know, my wife has red hair. And my previous 2 girlfriends before my wife also had red hair. So I really like redheads. So thank you. And that's the T-shirt.